Okay, so moving on here, what I want to do is talk about what the cell uses energy for and how these reactions relate to the use of energy in the cell. So the cell does three main kinds of work: mechanical, transport, and chemical. And we'll talk about these in just a second here. And all of these types of work are happen because of exergonic reactions that release energy, and so therefore will provide the energy to do this work. And it all involves a molecule called ATP. So cells manage energy resources to do work by energy coupling, they call it. So what in the world does that mean? It means that you're going to couple or come together two things. One reaction needs energy, one releases energy. So the one that releases energy provides the energy for the one that needs energy. So it says using a blank process to drive a blank one. So which one's going to release energy, ender or exergonic? It's going to be an exergonic, right? So an exergonic process releases energy and drives an endergonic one. So they call that coupling. They kind of work together. So this molecule called ATP stands for adenosine. Tri is the T. That means three. And what was there three of? Phosphate groups. So there's the P. And this is the cell's main energy source in energy coupling. So this says ATP is adenine plus ribose plus 3 phosphates. I just want to show you here that this is the adenosine molecule. And the adenosine triphosphate. Excuse me. So if we look at this, there's this right here is the adenosine molecule. And the adenosine molecule is made out of adenine. We know that from DNA. That's the A in DNA. And ribose, that's the sugar in RNA. All right, so it's in ATP. So this whole thing is adenosine made out of two smaller molecules put together. And then you have your 3 phosphate groups. So one here, here, and here. So there's 3 of those. 3 phos. 1, 2, 3. All right, your 3 phosphate groups. And that's the ATP molecule. So when bonds between the phosphate groups are broken by hydrolysis, remember, adding water to break down. So we're going to add water to break off a phosphate group. So we have 3, add water, breaks off 1. Add another water, breaks off another. All right, energy is released in this process. So when we break down ATP into ADP, which is 2 phosphates, if you take off a phosphate, plus a phosphate that's no longer attached to it, and we call that P sub I, as it stands for inorganic phosphate. It means it's not attached to anything. When we break that down, that is an exergonic process. All right, so that's exergonic, because energy is released. And this release of energy comes from the chemical change to a state of lower free energy. So the ATP molecule itself has more free energy than the ADP molecule. And so therefore, going back to this picture here, it's ATP being broken down into ADP and a phosphate, and both of those have lower free energy than the ATP. And so the energy is released. Oops, wrong way. Okay, and so so let's look at that. So this is like that. This is ATP being broken down into ADP in an inorganic phosphate, just like we just drew here, and that process releases energy. And so and that actually releases a certain amount of energy. So this is an exergonic reaction, and it releases energy, and the amount of energy is negative 7.3 kilocalories per mole. And so the negative means it's exergonic and releases energy. 7.3 is how much energy, and this is the units, kilocalorie. A kilocalorie is the same thing as what's called a big C calorie. So in real life, we have in real life, as if this is fake, a little C calorie. That's a unit of measurement of energy. Then we have a big C calorie, and that is 1,000 little C calories. And a big C calorie is the same as a kilocalorie. All right, and so that's what this is. So it's explaining how much energy there is. And so when we look at food labels and it tells you how much calories are in your food, they're actually measuring big C calories or kilocalories. So it's telling you how much per mole of ATP, how much energy you get, all right? So, or how much energy is released in that process. So we said that ATP is the main energy molecule. So how does it perform work? It does one of those 3 different types of work. And so the exergonic release of that inorganic phosphate is used to do endergonic work of the cell. So when ATP is broken down and energy is released, some of that energy is usable energy to actually do cellular work. So when ATP is hydrolyzed, it becomes ADP, right? And that's the adenosine diphosphate. So only 2 phosphates. And so that's what we see here. That's ADP right up here. All right, and so if we use ATP in this manner and break it down to get energy to do cellular work, can you see that eventually we might run out of ATP and have a surplus of ADP and a phosphate? And so what we have is a way to regenerate ATP so we don't run out of it. And so it's called the ATP cycle, and it looks like this. All right, so here we have our ATP, and ATP plus water, if we add water, that's this part right up here, we just add water, breaks off the phosphate group, and so you end up with ADP. So there's this part of the reaction. And this part, when ATP breaks down into ADP, this, let's do these in different colors here. That's this side. So this is an exergonic or endergonic reaction. This is an exergonic reaction, right? Because energy is being released. So energy is released for cellular work. So they're going to, so what is cellular work? Endergonic reactions are processes that need energy. And so that energy comes from the breakdown of ATP. On the flip side, if we keep on breaking down ATP, we're going to run out of it. So therefore, we have a way to regenerate the ATP or put the phosphate back onto ADP and go in this direction and remake ATP from the ADP and the phosphate that we broke down. And this process is an endergonic reaction because it takes energy to put that phosphate back on and to regenerate ATP. So it's showing you energy coming in. And so I want to talk a little bit about where that energy comes from to make ATP. So I'm going to write over here. I have more room on this side, but I'm going to keep the same color here. So let's just go over here like this. So let's write, where does this energy come from? And this comes from a process called cellular respiration, which actually is going to be our next chapter. It's the breakdown of glucose to get energy to make ATP. And this is called cellular respiration. So when we eat, we take in the glucose and we break it down, and we talked about that the breakdown of glucose is an exergonic reaction. So glucose being broken down into carbon dioxide and water. That's an exergonic reaction. It releases energy. Some of that energy is going to be used to put the phosphate back onto ADP to make ATP. Some of that energy is released as heat, so this is wasted energy. So therefore, the amount, the delta G here for cellular respiration, So the delta G for cellular respiration is negative because it's an exergonic reaction. So the negative just means it releases the energy. And how much energy? 686 kilocalories per mole. So that is quite a bit of energy that's released. So you can see up here that when you break down ATP, it releases 7.3 kilocalories per mole, which let's go ahead and add this to that. So in this case here, the delta G is negative 7.3 kilocalories per mole. So if that's the case, how many kilocalories per mole do you think it takes to make ATP? It's the exact opposite. It's positive 7.3 kilocalories per mole. So when I say that, where does the energy come from that we need to make every ATP 7.3 kilocalories per mole? Where that energy comes from is when we break down glucose in this process called cellular respiration. And you can see that every glucose, that per mole, we get negative 686 kilocalories per mole. So therefore, it only takes 7.3 kilocalories per mole to make one ATP. So can you see that mathematically, that each glucose molecule can make many ATP, all right, from that energy that is released. And so it's multiple ATP molecules. All right, and so then we use that energy to make ATP, and then it's the ATP that breaks down and drives our cellular work. All right, and so how does ATP drive cellular work? So these are three different examples here. So it says energy for cellular work. So it's, I'm gonna write here like these three things. All right, so what we have here, this is supposed to be, let me get a different color here, one that you can see better in here. All right, got me a pinkish color here. So this is supposed to be ATP. Whenever our book likes to use like little firecrackers, like showing lots of energy for ATP. And so it's showing you ATP is used for three different types of work, and it was these three types here: mechanical, transport, and chemical. So let's just go ahead and write what's going on here. So A here, this is supposed to be mechanical work. Things moving around. And I'm going to write here ATP phosphorylates motor proteins. Phosphorylate means adds a phosphate to. Phosphate group 2. And so phosphorylates it. So if you recall, this is a microtubule of your cytoskeleton, and let's say this is one of those transport vesicles, and the transport vesicle is going from the Golgi apparatus to the cell membrane, and how does it get energy to do that? So it phosphorylates this little motor protein right here, so I put here that that's a P, phosphorylates it, and then when the phosphate group comes off, the phosphorylation makes it less stable and more likely to react or move, and so therefore when it comes off, the little vesicle moves. All right, so that's mechanical work. We're going to see that the way ATP works is phosphorylating molecules. So they phosphorylate it, and in that process of phosphorylation and dephosphorylation, taking the phosphate group off, it causes work to happen. So this right here should look awfully familiar. This is active transport. And so ATP phosphorylates transport proteins. So I talked about this with the sodium-potassium pump. So that's what's going on here. Sodium-potassium pump, 3 sodiums out for every 2 potassiums in, and this transport protein is phosphorylated. Here's your phosphate group, and dephosphorylated, and causing that shape change, allowing sodium to go against their concentration gradient from low to high. And then lastly, we have chemical work. So this is like an endergonic reaction. And so ATP phosphorylates reactants. Okay, and so here are two molecules that are supposed to react, and so it phosphorylates one of the reactants and makes it less stable, more likely to react. All right, and so therefore, and then at the end you get your phosphate group over here not coming off, and therefore can be reused, and because when ATP gives up the phosphate, it's going to become ADP, and then that phosphate can be added back on to regenerate the ATP. All right, and so through cellular respiration, through the use of glucose. Alrighty, so let's move on here and take a look at enzymes now, and how enzymes play a role in this. So let me go back to my blue pen here. So a catalyst is a name, kind of a big umbrella term for any substance that can change the rate of a reaction without being altered in the process. So, it speeds up the rate of reaction, and so an enzyme is what we call a biological catalyst. And so it happens in living things. But there are other catalysts. So examples of a catalyst are enzymes. But also things that can speed up chemical reactions are heat, light, are things that can speed up a chemical reaction. An example of a reaction that can be sped up by these things is, the example is the breakdown of hydrogen peroxide. So this formula, hydrogen peroxide, is H2O2. So the formula, the balanced formula, is 2H2O2, gets broken down into 2 waters and oxygen gas. And so, so think about your hydrogen peroxide bottle. It's in a brown bottle. So that's the reason why, is because light can act as a catalyst for this reaction. So therefore, the brown bottle is stopping the light from getting in to get to the hydrogen peroxide and break it down. Because we want, when we buy hydrogen peroxide, we want it to be hydrogen peroxide. We don't want it to be water. And so therefore, light can do that. Also, if you read the bottle, it'll say to store in a cool place. And that's because heat can be a catalyst, all right, for this breakdown of hydrogen peroxide. And then in living things, what happens is that when you put hydrogen peroxide on a cut, it bubbles, right? And the reason why is because we have an enzyme called catalase. It's an enzyme that speeds up the breakdown of hydrogen peroxide. And so when you pour hydrogen peroxide on your cut, it bubbles. And what's in that bubbling is the oxygen, because it's breaking the catalase in your is breaking the hydrogen peroxide down into water and oxygen gas, and it's bubbling and so on. So that's an example of all 3 of these types of catalysts that can speed up a chemical reaction. So we're going to talk about enzymes, though. So we have sucrose here as an example, and we went through this in class about how sucrose breaks down into glucose and fructose. Do you guys remember if this is an exer or endergonic reaction? This is an exergonic reaction. So it's going to release energy, right? All right. But if you have sucrose is table sugar. So if you have table sugar sitting there, it's not going to just randomly start breaking down into glucose and fructose. All right, and let's talk about why. All right, so we have to have a reaction or something to help make sucrose more unstable, so that it is more likely that bond that's holding the glucose and the fructose together, that bond is more likely to break apart. All right, and so therefore be converted into glucose and fructose. So enzymes speed up metabolic reactions by by lowering what we call the activation energy, and this is the energy needed to start a reaction. So it lowers that activation energy. So And enzymes can do that. Heat can do that. Light can do that, and so on. By providing heat and light, provide energy that puts stress on the bonds, and therefore makes the bonds more likely to break, and therefore can be converted to glucose and fructose. And then enzymes can do that as well. So let's look at this particular graph here. So let's color code. So this is, we'll use orange here. All right, and so this says course of the reaction without an enzyme. And so the graph, let's just look at what we're looking at. This is the free energy, and this is the progress of the reaction. So the axes are the same as what we talked about with our bar graphs over here. So now this is a line graph. And so this is showing you right here where it starts is the amount of free energy in your reactants. And here at the end, this is the amount of free energy in your products. So you have more free energy in your reactants than your products. So this is a what kind of reaction? Energy is going to be released because there's more free energy here than here. Energy is released. And so this is an exergonic reaction. All right, so this is exergonic. Okay. So when we look here, the amount of free energy, so from the reactants to the products, that difference is the amount of free energy that's going to be released and be able to do cellular work. So here is the amount, that difference is the delta G. Now notice here we have a little uphill before so we have to, what does that mean? Free energy has to go up. What that means is that you have to have an input of energy in order to get those reactants to turn to product. All right, so that input of energy is called the activation energy. So think about this. So we just talked about sucrose. Sucrose is the two disaccharides hooked together, and they're sitting here, and they're pretty, and they're not going to break down. Even though breaking down is an exergonic reaction, they're not going to do that. They need a little bit of energy input to do that. But once that energy input comes in, then they can go ahead and, as it's made unstable, stress put on the bonds or so on, and it breaks down into two monosaccharides, then it can be converted to product, and you get that delta G. So E sub A refers to the activation energy. So this is E sub A whenever we see that. That's the activation energy. So you can see that there's activation energy needed to break the sucrose down and be converted to product. But notice here it says delta G is unaffected by the enzyme. So still, as we go up, we can't, when it goes back down and this energy is released, we can't include that in terms of energy available to do work because we had to input that energy. So this part right here doesn't count towards the free energy of the reaction. It's still only the difference between the reactants and the products is your delta G. And that's the amount of energy available to do work. Another example is gasoline. You have gasoline. If I threw a match in the gasoline, hopefully you could tell me that we'd have a big explosion. So it's a very exergonic reaction. And so, but gasoline, if you have a can of gasoline, it doesn't magically just combust and break down and have an exergonic reaction. What do you have to do? You have to throw a little match in it. That match is providing the activation energy. So the reason why activation energy is so important is because it stabilizes these reactants. Otherwise, they would always go to products, and we'd never have any of those reactants like gasoline on Earth because they would automatically be converted to product. And so therefore, it stabilizes it, and then once enough energy is absorbed and that molecule becomes unstable enough, then it can react and therefore convert to product, and then you get your release of energy there. So how does this relate to enzymes? So sucrose has an enzyme called sucrase. And sucrase, so whenever we see ASE, it is referring to an enzyme. So this is an enzyme. And the enzyme speeds this breakdown up. And so when we eat table sugar, sucrose, we have this enzyme that helps to break it down into glucose and fructose, two monosaccharides that we can use. So what it does is it speeds chemical reactions up by lowering the activation energy. So that is this part right here. Let me get my other marker here. So let's do this and color this one. Lowering the activation energy. All right, and so this is with an enzyme right here. So now, notice here, what they've done is that the enzyme has lowered the activation energy, so you don't need as much energy. So here's the sucrose, and it makes it so it doesn't have to absorb a lot of energy, and therefore causes the breaking into glucose and fructose a lot faster. So it increases that rate by lowering the activation energy. So the activation energy is lower with the enzyme. And we'll talk about, as we look into the next part here, how that actually happens. So let me do this.